Okay, that's enough. Так. Today we will discuss about information again. Information continues. Let's repeat what we know, what we have already known. We know that inflammation is general typical pathological process of local character. Yeah, local character, and it includes local damage of tissues and the local protective inflammatory reaction, which are response to this damage. Okay, inflammation formed in the evolution of the body's response to local injury, characterized by the alteration, disorders of the microcirculation with exudation and migration and proliferation, non-destruction and removal of the damaging agent from the body. Вот. Что еще можно сказать? Вспомнить. Inflammation is one of the most common types of pathological process. And at the same time, it is an important protective adaptive reaction. There are 3 stages of inflammation: inflammation, exudation, and proliferation. And there are 5 signs of inflammation. Fever, redness, swelling, laser, loss of function. Так. Так, что еще можно вспомнить? Etiology of inflammation мы можем вспомнить. Etiology of inflammation, there is a classification of etiology, that it causes inflammation of origin, endogenous and exogenous, and the causes of inflammation are nature, physical, chemical, biological, social, environmental factors. We know about conditions, some place of action, for example, or presence of local tissue damage or a certain number of microorganisms in the fabric, status of individual reactivity, sensitization, or strength and duration of action of the stimulus, and etc. Okay. Then let's remember about pathogenesis of inflammation. The pathogenesis of inflammation is a complex combination of neural, humoral effector mechanisms that underlie a large number of inflammatory phenomena constituting, in turn, effects and alteration with macrocirculation disorders, exudation, and immigration and proliferation. It's long story short. The role of tissue damage and inflammation. Что можно сказать? Okay, it's not today. Давайте теперь about mediators of inflammation. Да? More detail, так скажем. So the definition: mediators are the substances that initiate and regulate inflammatory reactions. There are cell-derived and plasma protein-derived inflammation mediators. There are vasoactive amines, lipid products, cytokines, products of complement activation. Okay, now I should lock our conference. Next, classification of mediators of inflammation. Is cell-derived and plasma protein-derived. Cell-derived, it means that mediators present in the cells, and plasma protein, it means that they are in the plasma. It's like complement activation, and factor Hedgeman factor 12 activation, kinase system, coagulative fibrinolysis system. This is the proteins, all of this C3A, C5A, C3B, 5B proteins, complex toxins. Okay, and cell-derived histamine, serotonin, prostaglandins, leukotrienes, play a activating factor, reactive oxygen species, nitric oxide, cytokines, neuropeptides, and this is the source of these substances. For example, histamine formed in the mast cells, basophils platelets, certain platelets, and the same other. What you can see in this diagram, a mediator, name of mediator, then source of this mediator, and actions. For example, histamine, source of histamine are mast cells, basophils, and platelets, and which actions, effects, are vasodilation, increased vascular permeability, endothelial activation. Next, serotonin. The source of serotonin is platelets. Next, actions, vasoconstriction, mostly vasoconstriction. Prostaglandins, mast cells, leukocytes. You can say that source of mediators is leukocytes because the mast cells and basophils, mast cells, for example, it's all types of leukocytes. You can see leukocytes leukocytes are source of more than 1, 2, or 3 factors. And actions, all of actions helps to progress, helps to information progress. Vasodilation, then vasoconstriction, again vasodilation, pain, fever, vascular permeability increasing, chemotaxis, it means leukocyte migration, leukocyte adhesion and activation, again dilation, increased vascular permeability, tissue damage, killing of microbes, reactive oxygen species, vascular smooth muscle relaxation, killing of microbes, local endothelial activation, systemic fever, metabolic abnormalities, hypertension. But besides cytokines, tumor necrotic factor, interleukin-1 and interleukin-6, they are causes of fever, secondary pyrogens. If you remember, when we discussed about fever, we I told you that cytokines are secondary parogens. Yeah, parogens are causes of fever, high temperature. Okay, and this is the systemic fever. Как раз то, что мы учили. Вот. Так. And chemokines, leukocytes activate macrophage is source of chemokines. So actions are chemotaxis, leukocyte activation. Okay. It was cellular mediators because sources of these mediators are cells. Then plasma protein derived. There are complement, kinase system, and proteases activated during coagulation. Because inflammation, during inflammation, this is the thrombosis. Yeah. The complement produced in the liver or так. Надеюсь, что я была здесь, да? So in the teleal activation, leukocyte recruitment. Так, что я говорила? Complement system, kinase system, and proteases activated during coagulation. There are all plasma protein derived mediators. They move to the plasma and they stay here. Stay in plasma. Так. Effects of complement are leukocyte chemotaxis and activation, direct target killing, vasodilation, so mast cell stimulation, and mast cell will produce this mediators. Yeah. Kinase, increased vascular permeability against mid-muscle contraction for the increasing vascular permeability, vasodilation, and pain. And this endothelial activation again and leukocyte activation. Active mediators are produced only in response to various stimuli like microbial products. Active mediators. Lifespan of mediators are very short. Yeah, they work, and after they will work, they will die. One mediator can stimulate the release of other, like complement activation, causes release of histamine and cytokines. It's like cascade. But the active amines, histamine and serotonin, acts on blood vessels. First mediators. There are first mediators of inflammation. After beginning of inflammation, histamine and serotonin are first who will help who will help. Now, mast cells are richest source of histamine. Yes, mast cells are source of histamine. Histamine stored as granules and released by degranulation and in response to various stimuli. Physical injuries, which stimulate them. Physical injuries, antibodies-mediated hypersensitivity reactions, allergy, and complement products, and toxins A3A, C5A, and for neuropeptides, substance B, and cytokines, interleukin 1, interleukin A. Histamine causes dilation of arterioles and increases the permeability of venules. It means that histamine cause of hyperemia, arteriolar hyperemia, hyperemia, and increases of permeability of venules. Okay. And the cause of swelling, histamine, principal mediator of the immediate transfer phase. It increased vascular permeability producing interendothelial gaps in venules, act via H1 receptors present on microvascular endothelial cells. Serotonin, performed mediator present in platelet and certain endocrinal cells like GIT. Primary function is as a neurotransmitter and vasoconstriction. Neurotransmitter. Arachidonic acid metabolites. No low fever. The lipid mediators prostaglandins and leukotrienes are produced from arachidonic acid, present in membrane phospholipids. Stimulate vascular and cellular reactions. Arachidonic acid derived from dietary sources or by conversion from the linoleic acid. Mechanical, chemical, physical stimuli, or other mediators release arachidonic acid from membrane phospholipids through the action of cellular phospholipases, mainly phospholipase A2. If you remember our diagrams when we were talking about fever, it was the diagram with mechanism of production of mediators of fever production, and arachidonic acid metabolites was present, especially phospholipase A2. Phospholipase A2 activation occurs via increase in cytosolic calcium and kinase. Allow me, students are waiting. Okay, okay, they can join. Because I see that internet connection is not good today. Talk and in response to external stimuli. You know that during cell injury, cytosolic calcium 2 plus increased very. This is the short scheme of effects of action of mediators of inflammation of mechanism of production mediators. What you can see, cell membrane phospholipids. After damaging, there is the phospholipases presence, and arachidonic acid will produce cyclooxygenase and 5 lipooxygenase. So cyclooxygenase COX-1 and COX-2. And this is the steroid drugs, and this is the aspirin, non-steroid, 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 non-steroid anti-inflammatory drugs, I think, like this. Aspirin and the tazine will inhibit cyclooxygenase. In this phase, will stop the inflammation process, and steroid on this phase when the phospholipases will act arachidonic acid. So there are two pathways of arachidonic acid metabolites: cyclooxygenase pathway and 5-lipoxygenase pathway. Так, что можно сказать об этом? Так, окей. So, this first pathway. Production of prostaglandins under the influence of prostaglandin, there are prostacyclin activation, thromboxane E2 production, and prostaglandin D2, E2, and there are causes of vasodilation, inhibits platelet aggregation, vasoconstriction, promotes platelet aggregation again, causes vasodilation, increased vascular permeability again. There are a lot of mediators who help to vasodilation and aggregation. And second pathway, 5-lipoxygenase pathway, it helps chemotaxis due to the mediators like trians. Effects of trians are bronchospasm, increased vascular permeability again, and will be swelling. You can see leukotriene receptor antagonist inhibits this pathway. And third, 12-lipoxygenase produce lipoxin E4 and lipoxin B4. Its effects is inhibition of inflammation. Okay. Prostaglandins. Prostaglandins. Тут есть у меня. Ладно, prostaglandins produced by mast cells, macrophages, endothelial cells. It involved in vascular and systemic reactions of inflammation. COX-1 and COX-2 cyclooxygenase is involved in synthesis of prostaglandins, as you can see. Well, cyclooxygenase pathway. COX-1 is produced in response to inflammatory stimuli and is also constitutively level expressed. COX-2 is induced by inflammatory stimuli. Again, platinum action, vasodilation, vasoconstriction, vasodilation, prostaglandins, and vasoconstriction, thromboxane A2, like increased vascular permeability, leukotrienes, and chemotaxis leukocyte adhesion, chemotaxis, which is endothelial wall, leukotriene B4 HEG reactions. Thromboxane A2, provided by action of thromboxane synthase present in platelets. Functions. There are platelets aggregation, vasoconstriction. It is unstable and converted to inactive forms. Thromboxane B2. Prostacyclin. There are all of cyclooxygenase pathway products. Prostacyclin formed by action of prostacyclin synthase present in functions increase vascular permeability, inhibit platelet aggregation. Formed by action of prostacyclin synthase present in vascular endothelium after damaging of vascular wall, vascular endothelium. There are a lot of mediators. Prostaglandin D2 produced by mast cells along with prostaglandin E2. It causes vasodilation and increases the permeability of postcapillary venules, thus potentiating edema formation. Prostaglandin Phe2 stimulates contraction of uterine and bronchial smooth muscles and small arterioles. Prostaglandins involved in the pathogenesis of pain and fever and inflammation. And prostaglandin E2 is hyperalgesic, makes the skin hypersensitive to painful stimuli. Leukotriene, produced by leukocytes and mast cells by the action of lipooxygenase. Lipooxygenase pathway. No. It involved in vascular and smooth muscle reactions and leukocyte recruitment. Three types of lipooxygenase we have. There are three types. First, lipooxygenase present in neutrophils. Neutrophils convert ketonic acid to 5-hydroxy-eicosapentaenoic acid. Act as chemotactic for neutrophils. For leukocytes, especially neutrophils, are cells of inflammation. Neutrophils. Leukotriene B4 chemotactic and neutrophil activator leads to aggregation and adhesion of the cells to venular endothelium. Generation of friction oxidation species, release of lysosomal enzymes. Leukotrienes leukotriene C4, leukotriene D4, and leukotriene E4 causes vasoconstrictions, bronchospasm, increased permeability of venules. Leukotrienes are more potent than as histamine in increasing vascular permeability and causing bronchial spasm. And that's why in this step we can use leukotriene receptor antagonist inhibitor, and we will blockage this reaction as bronchospasm. Lipoxins, generated from market and encased by the lipoxygenase pathway. It suppresses inflammation by inhibiting recruitment of leukocytes, inhibit neutrophil chemotaxis and adhesion to endothelium. Pharmacologic inhibitors of prostaglandins and leukotrienes, cyclooxygenase inhibitors. Cyclooxygenase inhibitors is the most important to you. The aspirin is the acetylsalicylic acid and other non-steroidal anti-inflammatory drugs. You can see it's inhibit both COX-1 and COX-2. COX-1 is responsible for the production of prostaglandins that are involved in both inflammation and homeostatic function. Prostaglandins that are involved only in inflammatory reactions, which homeostatic functions of fluid and electrolyte balance in the kidneys, site production in the gastrointestinal tract. And that's why aspirin is danger, aspirin danger and harmful for the gastrointestinal tract, especially for the stomach. It can be a cause of как это на английском было? Сейчас скажу. Formation of ulcer. That's why it's too important to use the cyclooxygenase inhibitors who will inhibit only COX-2. Lipoxygenase inhibitors, xylitol, corticosteroids, broad spectrum anti-inflammatory, reduce this genes encoding COX-2, phosphokines, and chemokines. Cytokines are proteins produced by many cell types, activated lymphocytes, macrophages, and dendritic cells mediate and regulate immune inflammatory reactions. Так, cytokines, да? Что у меня есть cytokines? So, the main source of cytokines, monokines, in inflammation are stimulated monocytes, and macrophages. Okay, activated lymphocytes, macrophages, stimulate monocytes. These polypeptides are produced by neutrophils, lymphocytes, endothelial, and other cells. Further, the most studied cytokine is interleukin-1 and a humor necrosis factor. Cytokines increase vascular permeability, network-dependent, but the adhesion and immigration of leukocytes, in addition to the pro-inflammatory properties of cytokines, may play a role in the direct protection of the body, stimulating the neutrophils and monocytes to killing the absorption and digestion of microorganisms, as well as stimulating the neutrophils and macrophages to kill. Так. And what еще можно сказать еще? Stimulants for secretion, there are microbial products, immune complexes for bodies, physical injury, trauma, and variety of other inflammatory stimuli. And again, the imaging of endothelial cells. TNF production is induced by TLRs, and other microbial sensors. This is the receptor of microbial microbial membrane. This is the cytokines, source of cytokine in principal action. Очень very bad. Очень плохо видно. Unvisible. Roles of cytokines in inflammation. First, endothelial activation by tumor necrotic factor and interleukin 1. Increased expression of adhesion molecules, P-selectins and ligands for leukocytes, and P-selectin and E-selectin and ligands. They are molecules or receptors for the leukocytes receptors, which helps to process of adhesion of leukocytes. Increased production of various mediators, increased procoagulant activity. Activation of leukocytes and other cells stimulates microbicidal activity of macrophages by inducing production of NO, nitric acid. Interleukin 1 activates fibroblasts to synthesize collagen and stimulates proliferation of synovial and other mesenchymal cells. Simulates Th17 responses induce acute inflammation. Systemic acute phase response induced by interleukin 1, TNF, and interleukin 6. TNF regulates energy balance by promoting lipid and protein mobilization, and by suppressing appetite leads to cachexia. So very low weight. TNF antagonists have been remarkably effective in the treatment of chronic inflammatory diseases, rheumatoid arthritis, and psoriasis. Roles of cytokines. You can see this is the vascular wall, endothelial cells, and basal membrane. This is the basal membrane. What is the basal membrane? And this is the cells, endothelial cells. This is the nucleus. You can see local inflammation. Why local inflammation? Because we have systemic inflammation, yes, also, but we are talking about local inflammation of vascularized tissues after damaging. So, what you can see, increased expression of adhesion molecules. There is the P-selectins, tumor necrotic factor, interleukin 1, tumor necrotic factor, ACT, and increased expression of adhesion molecules for the leukocytes. So TNF and interleukin 1 activates leukocytes, and leukocytes produce interleukin 1, interleukin 6 chemokines, what you can see, and increased permeability by interleukins. Systemic protective effects. Ладно, это не так уж важно. Protective effects. Brain fever, hypothalamic. Как его? Hypothalamus, да? Interleukin 1, interleukin 6. Acute phase proteins, liver products acute phase proteins, and under influence of the interleukin 1 and 6, bone marrow start products leukocytes better. Okay. Systemic pathological effects. Heart under influence of TNF. What happens with heart work? Low output. What happens with endothelial cells and blood vessels? Thrombus, and increased permeability. Multiply tissues. Under influence, skeletal muscles, insulin resistance. What is the insulin is, yeah, helps to glucose. Так, chemokines, да? Так, what about chemokines? We can see. One. Okay. Chemokines are a family of small proteins that act primarily as chemoattractants for specific types of leukocytes. About 40 different chemokines and 20 different receptors for chemokines have been identified. Classification of chemokines. Chemokines, yeah. Hello to enter the class. What happened again? Запарили уже. Four major groups of classification C chemokines have one amino acid residue, separating the first two of the four conserved cysteine residues. Act primarily on neutrophils secreted by macrophages in endothelial cells. Function activation axis of neutrophils. Interleukin function. Stop 6C chemokines have the first two conserved and sustained residues adjacent. It includes monocyte chemoattractant protein, monocyte chemoattractant protein 1, eotaxin, macrophage inflammatory protein 1 alpha, veins regulated normal T cell expressed and secreted, attract monocytes, eosinophils, basophils, and lymphocytes. Eotaxin selectively recruits eosinophils. And C chemokines, like the first and third of the four conserved cysteines, except lymphotactin, specific for lymphocytes. So this was C and C3C chemokines contain 3 amino acids between the two cysteines. This is the biochemistry, and you couldn't memorize this. Fractalkine exists in two formats. It's not as matter for us. Cell surface bound protein promotes strong adhesion of monocytes and T cells. So this is the important. Soluble form derived by proteolysis of the membrane bound protein. Potent chemoattractant. Chemokines mediate their activities by binding to 7 transmembrane G-protein coupled receptors. G-protein coupled receptors. Functions of chemokines. This is important. In acute inflammation, stimulate leukocyte attachment to endothelium by acting on leukocytes to increase the affinity of integrants. Chemotaxis, moving leukocytes through the vascular volume. Maintenance of tissue architecture. Homeostatic chemokines produced constitutively in tissues. Other cytokines in acute inflammation. Interleukin 6, interleukin 17, type I interference, secreted by macrophage interleukin 6, involved in local and systemic reactions. Interleukin 17 produced by T lymphocytes promotes neutrophil recruitment. Type 1 or type I think, concerns some systemic manifestations. Complement system. Complement system, this is the plasmic, yeah? Mediator. The complement system is a collection of soluble proteins and membrane receptors that function mainly in host defense against microbes and in pathologic inflammatory reactions. The system consists of more than 20 proteins, some of which are numbered C1 through C9. Functions in both innate and adaptive immunity. Innate immunity, adaptive immunity. The activation and functions of the complement system. Окей. Так. Это очень плохо видно, да, again? There are two pathways. There are three pathways. One pathway, first classical pathway and lectin pathway. Uninvisible. Okay, this is the effects, destruction of microbes by leukocyte, phagocytosis of microbe, and lysis of microbe. The effects depends on the pathways. First pathway, alternative pathway, classical pathway, and lectin. Okay, microbe. This is the microbe. Yeah, microbe. This is the microbes. Ничего не вижу. Binding lectin, mannose binding lectin. This is the mannose binding lectin. And it's the antibody, V cells. What happens? C3b is deposit on microbe. Вообще не вижу ничего. В общем, what you can see C3, C3A, C3b. This is the complement system proteins which consist of complement system, and which effects of this? What is the effect of each of these pathways are destruction of microbes, okay, phagocytes. So, давайте здесь посмотрим, может быть, здесь есть механизм. Complement proteins are present in inactive forms in the plasma. This means that this complement protein mediator is plasmic, the plasma-derived information of plasma-derived mediator. Present in inactive form. The critical step in complement activation is the proteolysis of the third. Cleavage of C3 can occur by one of three pathways. This is the explanation of this scheme. Okay, let's try to understand. Classical pathway, okay, triggered by fixation of C1 to antibody immunoglobulin M or immunoglobulin G antibodies. This is they are antibodies that has combined with antigen. Okay, triggered by fixation of C1 to antibody that has combined with antigen. Then the C3 C1 to antibody globulins. Then alternative pathway. Нет, the classical pathway. Вот они, antibodies, да? Immunoglobulin MRG. Вот они. Triggered by fixation of C1 protein that has combined with antigen. Okay, alternative pathway triggered by microbial surface molecules and a toxin or lipopolysaccharide lipase. complex polysaccharides, cobra venom, and other substances in the absence of antibody. Okay, absence of antibody. Absence of antibody. What you can see, microbe antigens with lipopolysaccharides, maybe. The lectin pathway plasma nodes binding. The lectin binds to carbohydrates on microbes and directly activates C1. Okay, these all pathways are activate C1 complement, yeah? But this is mono binding lectin. Хорошо. Functions of complements. Inflammation, C3, C5a, and C4a. Stimulate histamine release. They are called anaphylatoxins. Stimulating histamine release, they are called anaphylatoxins. Don't forget, anaphylatoxins, they are complement system proteins, C3a, C5a, and C4a. They stimulate histamine release. C5a, chemotactic neutrophils, monocytes, eosinophils, and basophils. Opsonization and phagocytosis, C3b and IC3b. Oxidation and phagocytosis are effects of C3b and IC3b. Action cell lysis, the position of the MAC on cells. MAC shortly, конечно. On cells makes these cells permeable to water, permeable to water and ions, and results is death lysis of the cells. Membrane attack complex. Okay. Cell lysis. So yeah, I think you can understand. The activation of complement is tightly controlled by cell-associated and circulating regulatory proteins. C1 inhibitor, this is the proteins, is regulating regulatory proteins. C1 inhibitor blocks C1 activation. Inherited deficiency of this inhibitor is the cause of hereditary angioedema and edema. Decay, accelerating factor. Decay accelerating factor and the CD59. DF inhibit seed-free convertase formation. Decay accelerating factor. Yeah. CD59 inhibits formation of the membrane attack complex, and acquired deficiency leads to PNH. Так, давайте, значит, еще раз повторим. Так, activation of complements controlled by regulatory proteins, cell-associated regulatory proteins. C1 inhibitor block C1 activation, decay accelerating factor, and CD59. Decay accelerating factor inhibits C3 convertase formation, and CD59 inhibits formation of the membrane attack complex MAP, yeah? The membrane attack complex. And acquired deficiency leads to PNH, paroxysmal nocturnal hemoglobinuria. Other mediators of inflammation. Platelet activating factor, phospholipid-derived mediator, secreted by platelets, basophils, mast cells, neutrophils, macrophages, and endothelial cells. It causes platelet aggregation. You can see it's the same name, platelet activating factor, platelet segregation, yeah. Vasoconstriction, bronchoconstriction at low concentrations, it induced vasodilation and increased venular permeability in low concentrations. Products of coagulation. Kinins, vasoactive peptides derived from plasma proteins called kininogens by the actions of specific proteases called kallikreins. Sorry. So this is the vasoactive peptides from which formed from kininogens under the influence of plasma kallikreins, non-peptide bradykinin, for example. The trigger factor activation kallikrein kinin system is activated when tissue damage, Hegeman factor, 11 coagulation factor, prekallikrein, and converting kallikreins. The bradykinin increases vascular permeability and smooth muscle contractions, dilation of blood vessels, and pain. Bradykinin is cause of dilation of blood vessels and pain. Nitropeptides, the crida is secreted by sensory nerves and various leukocytes. Substance P, there are substance P and neurokinin A. Substance P prominent in nerves, fibers, in lungs and gastrointestinal tract. Functions: transmission of pain signals. Its first function, then regulation of blood pressure, stimulation of hormone secretion by endocrine cells, increasing vascular permeability. So trigger factor activation, this system, kinin bradykinin system is Hegeman factor. Hegeman factor is 11 coagulation factor. And kinins mediate the expansion of arterioles and venules by increased permeability of endothelial cell contraction. They cut the smooth muscles of the veins and increase intracapillary and venous pressure. This is why regulation of blood pressure. Kinins inhibit immigration of neutrophils, macrophage distribution, stimulate mitogenesis and migration of T lymphocytes and secretion of lymphokines. They enhance proliferation of fibroblasts and collagen synthesis, and consequently may be important in the pathogenesis of repair phenomena of chronic inflammation. One of the most significant effects of kinins activation of reflexes by stimulating sensory nerve endings, and this endings gives rise to inflammatory pain. Kinins cause of enhanced histamine release from mast cells, prostaglandin synthesis in many types of cells. So that's why they can be cause of vasodilation, smooth muscle contraction, and pain is associated with release of other mediators, in particular prostaglandins. And activation of Hegeman factor triggers not only the protein formation, but coagulation and fibrinolysis also. Used for mediators such as fibrin peptides and fibrin degradation products, which are potent chemotrectin. You know that fibrinolysis and clot formation in blood vessels focus are significant as in the pathological and predictive inflammatory phenomena. Morphologic patterns of acute inflammation. The morphological hallmarks, dilation of small blood vessels tissue. Various morphological patterns occur: serous inflammation, fibrinous inflammation, purulent inflammation, abscess, ulcers. It's morphological patterns. This is the pathology anatomy. I think it will be So today is enough. This is pathanatomy start.